I think I already showed you a picture of it. It's better than me, for sure. Younger. He's, what? How old are you? He's 26. It's not fair. But I have to warn you, I don't think it's social. All right, please. Please be seated.
And then I open this academic ceremony, in which Shan Wang will defend her academic thesis called The Role of Diet in Inflammatory Bowel Disease, a focus on the underlying mechanisms via aryl hydrocarbon receptor signaling. Before we go there, to the defense, I would like to introduce the supervisors, because otherwise they're never named. And the supervisors are Professor Frederik Jan van Schoten. He's a professor of genetic toxicology. Professor Daisy Jonkes, professor of intestinal health. And Dr. Roger Godschalk, associate professor in genetic toxicology, all of our university. My name is Nalle de Vries. I'm replacing the rector. And having said all this, I will announce the other people present here or online when it's their turn to ask a question. But first, I ask the candidate to give a short presentation of her work. The floor is yours.
Okay, thank you. So highly esteemed the prorector, the members of Corona, the supervisors, the dear colleagues and friends, thanks for being here today. So in the upcoming 15 minutes, I'm going to present the summary of my thesis entitled The Role of Diet in Inflammatory Bowel Disease, a focus on the underlying mechanism via aryl hydrocarbon receptor signaling. So the inflammatory bowel disease is chronic inflammatory disorders of the gastrointestinal tract. It contains two subtypes, the ulcerative colitis and the Crohn's disease. The highest prevalence of IBD is observed in the Western countries, while in the newly industrialized countries, the incidence of IBD is increasing rapidly from the 21st century. So the IBD is characterized by alternating causes of quiescent remission and active intestinal inflammation, which we also call it flare. So the flare is really difficult to predict. And without timely and effective treatment, the disease will progress and develop severe complications, such as the bowel obstruction, fistulas, malnutrition, and even colorectal cancer. So the current treatment aims to induce and maintain prolonged remission and avoid disease progression. So the cause of IBD still remains largely unknown, but it is widely accepted that the IBD results from a complex interaction between the environmental, microbiome, and the immune-related factors in a genetically susceptible host. So among the environmental factors, the diet seems to play a very important role in IBD development. For example, the people who stick to a Western type of diet tend to have a higher risk of IBD development. On the contrary, adherence to a Mediterranean diet is associated with a lower risk of IBD development. So definitely the people want to ask, so what to eat to prevent the IBD development and also progression? To answer this question, it's very important to know the underlying mechanisms, how diets influence the gut microbiome and immune response. So in this thesis, we explore one possible mechanism through the modulation of the aryl hydrocarbon receptor, AHR. So the AHR is a ligand-activated transcriptional factor. With the ligand binding, the AHR will translocate from the cytosol to the nucleus and regulate the gene expression. And in the gut lumen, many metabolites from the food intake or from the microbiome metabolism were reported to be AHR ligands. And with the ligand binding, the AHR activation in the intestinal epithelial cells and immune cells will help to maintain the barrier integrity and also regulate the immune response in order to prevent the invasion of the pathogens. But how the dietary AHR ligands were changed in IBD and their relationship with the IBD development still need to be identified. So in this thesis, we aim to explore the role of different dietary factors in the development of IBD and subsequent risk of developing complications. In addition, we want to identify the role of dietary AHR modulators in IBD development in order to identify novel diagnostic, preventive, and therapeutic targets. So the first study, which is the chapter of the book, we look into the role of diet in genotoxicity of fecal water. So the colitis-associated colorectal cancer is one of the most severe complications of IBD. So the chronic inflammation in the intestine will induce the release of free radicals and also the genotoxic microbiome metabolites in the gut lumen, and these compounds will induce the DNA damage in the chronic epithelial cells. So the accumulation of the DNA damage in the chronic epithelial cells will induce the formation of dysplasia and further proceed to carcinoma formation. So this study is a cross-sectional study, and we collect the fecal samples from IBD patients and the healthy controls, and we isolate the fecal water, and we use the fecal water to treat the chronic epithelial cells and measure their genotoxic potentials in the chronic epithelial cells. In addition, we also have their clinical data, food intake data, and then we analyze the association between the genotoxic potential of the fecal water, the food intake, and also the intestinal inflammation. So the results show that the food groups, including soy products, coffee, the dietary pattern rich in potatoes, white meat, nuts and seeds, eggs, legumes, and the soy products, seem to reduce the genotoxic potential of the fecal water. However, on the contrary, the food groups, including the processed meats, sugar, cakes, and the confectionaries, the nutrients, including the added sugar and hand iron, is positively related to the genotoxic potential of the fecal water. And then chapter 3, we investigate the effect of protein fermentation products in gut health. So with more protein being intake from the food, the more undigested protein will go into colon and be fermented by the gut microbiota and produce various microbiome metabolites. And these metabolites will definitely influence the function of the intestinal cells. So in this study, an in vitro model of the human distal colon was applied. And the high protein diets with 3 different protein sources, including casein, vital wheat gluten, and lentil, was submitted into the system. And then we measure the production of the microbiome metabolites. We also collect the lumen samples, and we isolate the aqueous phase, and we use the luminal extractions to treat different kind of in vitro cell models. And the results show that the fermentation of high lentil protein diet induce the production of more beneficial microbiome metabolites. In addition, the luminal extractions from the lentil fermentation induce less damage to the barrier integrity. In addition, it also induces less cytokine secretion by the macrophages. And the chapter 4 and 5, we investigate the intestinal tryptophan metabolism in IBD. The tryptophan is an essential amino acid, and the main source of tryptophan comes from the food intake. And the tryptophan can be metabolized in the gut through 3 different pathways, including the kynurenine pathway in the intestinal epithelium and the immune cells, the indole pathway in the gut microbiota, and also the serotonin pathway in the enteroendocrine cells. And most of the tryptophan metabolites were reported to be AHR ligands or AHR modulator. So the chapter 4, it's still a cross-sectional study, and we use the same population as we used in the chapter 2. And we still have their fecal samples. We measure the fecal calprotectin level to indicate the presence of intestinal inflammation. And we also isolate the fecal water, and we measure the AHR activation ability of the fecal water. In addition, we also measure the concentrations of the tryptophan metabolites in the fecal water. So the results show that the activation of the kynurenine pathway is positively related to the intestinal inflammation, while negatively associated with the AHR activation ability of the fecal water. While the activation of the serotonin pathway and also the formation of indoleacetic acid in the microbiota were negatively associated with the intestinal inflammation, while positively associated with the AHR activation ability of the fecal water. And for the next step, we further explore how AHR activation ability of the fecal water can be explained by the tryptophan metabolites. So what we do is we treat the colonic epithelial cells with the fecal water, as well as the mixture of tryptophan metabolites with the concentrations that determined in the fecal water. And then we find the mixture of tryptophan metabolites did not induce very obvious AHR activity when compared with their original fecal water. So definitely tryptophan metabolites are not the main AHR agonist in the fecal water, and the major agonists still need to be identified in the future. And in chapter 5, we identified the involvement of intestinal tryptophan metabolism in IBD by integrating so far published transcriptome and the Mentablon studies that compared between IBD patients and non-IBD control. And through integrating the transcriptomic and the metabolomic data, we found the decreased tryptophan absorption, enhanced the kynurenine pathway, increased interstitial serotonin availability, and altered the indole pathway in gut microbiota in the IBD patients, especially in patients with active disease. And the chapter 6, it's a review. And then we summarize the so far reported dietary AHR modulators and their possible concentrations that present in the intestine. And we further summarize the underlying mechanisms, how AHR activation maintain the intestinal homeostasis through regulating different kind of intestinal cells as well as the gut microbiota. In this review, we further discuss the potentials and challenge of applying dietary AHR modulators as a target for IBD management. So, to conclude, based on the findings of my studies, the intakes of certain dietary factors are associated with the genotoxic potential of the fecal water, which could help to develop potential preventive dietary interventions for IBD patients. And the second, the influence of different protein sources on gut health should be considered when a high-protein diet is consumed. And third, the fecal water from IBD patients induced lower AHR activation. The major AHR ligands in fecal water still need to be identified. And for the aberrant intestinal tryptophan metabolism is present in IBD patients. And lastly, the mechanistic work incorporating omics approaches may enable a more accurate delineation of individualized nutritional response. Thank you for your attention, and I will give back the floor to the prorector.
Yes, thank you, and thank you for the presentation. Of course, I forgot to introduce your two paronyms, and that's well, you know, I'm trying to make up now. So Wenbu Wu and Yingji Wu, welcome too. And now we'll start the actual defense, and the first question will be asked by Professor Blaak, and she's a professor of human biology of our own university.
Thank you, Mr. Prorector. Dear candidate, first I would like to congratulate you with this very nice, well-written thesis, which I read with great pleasure, and also I'm impressed by the broadness of approach, going from controlled in vivo experiments you execute, as well as going to a review and a meta-analysis and systematic review of the transcriptome and metabolome, so a little bit going from cell to more complex data sets. And of course, I'm here to exchange thoughts on a few of the issues. I would like to start off with chapter 3, with respect to the TIM2 experiments. And you compare the different protein sources. What I was just wondering, first, a few questions on the setup of the experiments. Did you consider also to include a pre-digestion step, a batch fermentation or TIM1 model when looking at the effective proteins?
Highly esteemed the opponent. Thanks for your kind words and also your question. Yeah, I think definitely I agree with you for the effect of protein in the gut health. Actually, it's also very important to include the digestion, absorption in the upper gastrointestinal tract. But I think that would be like the next step. And then you can combine with, for example, as you mentioned, the TIM1 sample, which is the in vitro model of the small intestine, combined with the like the cell models to analyze the absorptions, and by using like in silico computational models to predict the protein absorptions by the cells.
Okay. Yeah, I agree, but you didn't do that pre-digestion step in the current model. How does that possibly affect your short-chain fatty acid concentrations you measured? Do you think there's an impact?
You mean, sorry, so you mean like whether the digestion and absorption will influence the production of the short-chain fatty acids?
Yes.
So I have to say that because when we say the protein fermentation, actually we aim to the undigested protein. So it's not the digested protein. So yeah, because we mentioned like the with like a lot of protein being intake, like more undigested protein will go into colon.
Yeah, I fully agree, but you didn't take that now into account because you added the protein immediately to the colonic model.
Yeah.
I'm just wondering, but every experimental setup is different. But when I compare the cumulative short-chain fatty acid production after your protein, in particular the lentil protein, it seems to be rather high. I always had the impression proteolytic fermentation gives a little bit lower short-chain fatty acid concentrations, but...
Oh, but I still need to specify like the diet that we submitted into the TIM2 model. Actually, it's the standard Elian efflux median plus additional protein sources. Mm-hmm. So the standard Elian efflux median mimics the terminal like contents, like the contents from the terminal Elian. So actually, this part actually mimics like the content from the small intestine digestion. Yeah.
Yeah. Okay. I was just wondering on the duration of the experiment, 72 hours. Is that standard, or why did you choose for such a long period to follow the production of metabolites in the TIM2 model?
So you mean like the whole fermentation incubation in the it's like 24 hours, right? I think it's like I think we have like 12, like 24, 48, 72
Okay.
hours. We have like a timeline.
Yeah, okay.
Yeah.
That's correct. Thank you for
Yeah.
elucidating that for me. And then
Yeah.
I go a little bit farther, and as you also indicated in your nice presentation, then the lentil protein seems to be very positive, and not only with respect to short-chain fatty acid production, but also other effects on barrier integrity, IL-6 production in macrophages. When I extrapolate that a little bit to dietary guidelines, and you refer to that in your discussion, the Aspen guidelines, that protein intake has to be higher. It's 1.2 to 1.5 gram per kilogram body weight. Would that then mean that we in particular have to increase plant-based proteins because these seem to have more beneficial effects? What is your view on that?
Yeah, I think that's a very interesting like topic we need to explore for the next step, because like in the current results that we started actually were based on like a healthy colon situation, right? Like all these effects of the protein fermentation products, like were established in a healthy model. But you know, to give the protein suggestions for IBD patients, you need to take into the inflammatory status of the intestine into account, right? So I would say I think it's very meaningful to explore that.
Yeah, and just to proceed a little bit on that, because you indicate there's a loss of lean body mass also in patients. And when you look at gastrointestinal health, perhaps plant-based is a little bit more positive, but what in view of lean body mass?
Yeah, then you have to consider about like the efficiency of the proteins, right? Yeah, because when you want to give protein suggestions, actually you need to consider different aspects of the protein intake. Not only the effect of the protein products on the gut health, but you also need to consider about their like their efficiency in muscle gain, right? So I think for the next study, you need to find a balance between these two in order to have a better muscle gain as well as decreased gut health effects.
Okay, thank you. A final last brief question, because you refer several times to precision nutrition, and or more personalized responses to diet. And of course, then I'm wondering, what would you focus at first, or on what topic would you focus? It can be, for instance, production of short-chain fatty acids, which may be important. It can relate to the genotoxicity of fecal water or microbial composition. So what would be your approach in that?
Yeah, I think talking about the individualized nutrition or personalized condition, I think there are like, in my knowledge, I think there are three elements, like three very primary elements. Is the first is you need to give, you need to know like the exact status of the individual's the status. For example, like their gut health, and then like their body condition, whether they have like a chronic disease or something else. And then the second is what is the goal of your like your dietary suggestion or dietary interventions, right? And the third is you need to always follow like the feedbacks of the individuals after giving a dietary intervention. And based on my study, I think, like for example, like you monitor the tryptophan metabolites production in the gut, and also measure the genotoxicity of the fecal water. And also measure the AH activation ability of the fecal water would be a good indicator to monitor the status of your gut health.
Okay.
And also, of course, to monitor like the response after the dietary intervention.
Okay, thank you very much. It's nice to discuss that a little bit further, but now I give the word back to the prorector.
Yes, thank you, Professor Black. For the next question, I welcome an online guest. Professor Jerry Wells is a professor of host microbe interactions at Wageningen University, and we see him on the screen.
Thank you, Honourable Pro-Rector. Dear candidate, I enjoyed very much reading your thesis, and it got me thinking about dietary interventions in many diseases and effects on the epithelia and immune system. I would like to ask some questions about chapter 3, where you took the fermentation products from the TIM2 model, and after centrifugation of these, applied them to cells to study the effects. And my questions are about what we can really interpret and how we interpret information from those experiments. So I think you used a percentage of the fermentation products and added it to the cells. And do you have an idea how high the concentration of the short-chain fatty acids were, given the percentage of that?
Highly esteemed the opponent, thanks for your kind words and your question. Yes, we measure the concentration of the short-chain fatty acids in both the lumen samples as well as the dialysates. Yes, we do know the concentration of it.
And what concentration would have been applied to the cells?
I would say in this study, we didn't treat the cells with the concentrations, with the short-chain fatty acids, with the concentrations that determined in the samples. But with the study in the chapter, I think it's 4, actually we treat the cells with the short-chain fatty acids with the concentrations that we measured in the fecal water. Yeah. No.
But did I understand this wrongly then, that the fermenter from the TIM2, that does contain short-chain fatty acids, right? Because it includes the microbiota, which will ferment and produce short-chain fatty acids, or not?
Sorry, could you rephrase your question?
Yeah, the fermentation products that you put on the cells from the TIM2 model in chapter 3, it contains AHR agonists, but it also contains short-chain fatty acids, correct? Or not?
Like the AHR will induce the short-chain fatty acids?
No, I'm just thinking about the effects of short-chain fatty acids as well, because it has been reported that they enhance AHR activity in cancer cells.
Yeah, I think so. And actually, I discussed the effect of short-chain fatty acids on the AHR activation in the discussion of the chapter 3. Yeah.
And what did you conclude there?
Yeah, I think for sure, like the short-chain fatty acids, they have the impact on the AHR activation. But like the high concentration of the tryptophan and also high concentration of the isobutyrate, they actually could work as a AHR antagonist of the on the AHR activation, right? And then there's a mixture of these compounds, right? And then like the final AHR activation could result from like the interact reaction of all these compounds. Yeah. So
Okay.
Yeah.
Okay, thank you for that. I would also like to know about the experiments with the macrophage cells, the monocyte-derived macrophage cells from THP1 cells. So could you remind me exactly what you added to those cells to study the effects of AHR in chapter 3?
Yes, so we differentiated the THP1 cells from the monocyte to the macrophage. For the macrophages by the PMA, sorry, I couldn't remember the full name, for like the PMA for 48 hours. And then after 48 hours, we remove the PMA and then we rest the cells for 24 hours. So the reason why I did that is we find after the PMA induction, like actually the baseline of the cytokine increased quite a lot. So
yeah. And how did you test the effect? What did you do with can you explain how you used the fermentation product? So it was a dilution when you added it to the THP1 cells and then measured cytokine expression. Is that correct?
Yeah, that's true. Yeah. So that's why we just like differentiated cells and then we by the PMA and then we rest the cells for another 24 hours and then the baseline of the macrophages will decrease and to more common levels. And then we treat with the luminol extractions from the different protein fermentation products.
Yes. And will those fermentation extracts also include short-chain fatty acids?
Yes.
Yes, because they have effects on immune cells as well. I mean, it's quite well known that butyrate, for example, will suppress cytokine activity. So if the different fermentation products coming from different protein sources in the TIM2 contain different amounts of butyrate, for example, this could influence the response.
I totally agree.
I don't know if you're familiar with that literature. Some of it's fairly recent, but even at low concentrations, they affect cytokine production by immune cells.
But I think like the production of the short-chain fatty acids, it's quite consistent with the lower cytokine secretion by the lentil protein fermentation products. So I would say maybe the short-chain fatty acids play a role in a lower cytokine secretion, but definitely in this chapter we only explore the role of tryptophan metabolites. But I think I totally agree with you. The short-chain fatty acid may also play a role inside, but I mean maybe we didn't. So I think that's the drawback of my of this study.
I think untangling that is complicated unless you test the metabolites individually at the concentrations from the extracts and the short-chain fatty acids, etc. So that it gets very difficult. Yeah, and it's the same with CACA2 cells. I think that. Because it's an interplay between many factors, but it's challenging. But I think you certainly raised a lot of questions for us. I just have one more quick question, which is about the effects of AHR beyond the gut. I mean, you've been studying epithelia, but in the literature, it appears that the induction of IL-22, which has profound effects on the epithelium, barrier function, etc., is one of the most important effects of AHR activation. So that means the AHR metabolites are going beyond the gut epithelium and reaching the innate lymphoid cells that type 3 that produce this IL-22. Do you think that would be something worth pursuing in the future?
Sorry, so what is the kind of compounds you mentioned?
They are lymphoid cells that produce IL-22, and there are receptors on the epithelium that respond to IL-22, and it profoundly affects the barrier function, production of antimicrobials, which modulates the microbiota, etc. So I just wondered if you thought about that as a mechanism and whether that could be pursued also in vitro in some way.
So
or studied in vivo.
You are talking about like a receptor on the chronic epithelial cells, right? Yeah, I think.
Yeah.
Yeah, definitely. Maybe even though I didn't look into this, but I mean there are many mechanisms that underlying the like the interaction between the foods, the gut microbiome, and the immune system. The AHR is just like one of the mechanisms, and maybe the AHR is not the main mechanism, but I mean we still need to identify that, right? And if it's not important, and then we can say it's not important. But if it is, and then it would be a very interesting like targets for the IBD management.
Okay, thank you very much. I think I've run out of time, so I pass the word back to the Honourable Rector.
Thank you very much, Professor Wells. And I turn to another guest from China in this case, so you're very welcome as well. Professor Wen Chen is a professor of epidemiology at Sun Yat-sen University in China, your alma mater. The floor.
First of all, congratulations for the accomplishment of this thesis. Sounds a great job, and I enjoyed to read the thesis and appreciate the research design and the method lines in the food, utilization of patient samples, integration of in vitro and in vivo experiments and elucidation the interaction between metabolic abnormalities and the gut microbiome in maintaining intestinal cell homeostasis and its regulatory mechanism. Well done. So I have two questions. Question one is regarding the chapter in chapter 2. So why is genetic toxicity chosen as an important indicator to assess disease status and the impact of dying? Have other indicators such as cell toxicity or oxidative stress being compared in experiments? In addition, in the study design, the severity of a patient's condition or the progression of the disease may be significant influencing factors. So during the experiment, what's your quality control in place regarding this aspect?
Highly esteem the opponent. Thanks for your kind words and your question. I think we didn't use we didn't measure the reason we measure the genotoxicity of the fecal water is we want to predict the risk of inflammation-induced dysplasia. So actually, we didn't measure this. We didn't do these experiments to predict the disease status. That's for one thing. And the second is, yeah, definitely. I mean, during the process of the inflammation-induced dysplasia and the carcinoma formation, there are many cell events happening for not only the DNA damage, but maybe also like oxidative stress and maybe like the blockage of the cell cycle, the apoptosis. I mean, definitely, if you can measure all these cell like experiments to indicate the fecal water activity, that will be the best. But and I would say, if you really want to do all this, like the transcriptomic analysis of the cells treated by the fecal water would be a very nice method. But the point is, it's quite expensive, and also, I mean, we have like big populations, right? So I think based on our current situations, I think it's nice to start with like a relatively simpler, nice indicator and like experiments and like the DNA damage. It's widely thought to be an initial events, initial events for the dysplasia formation. So that's the one reason. And the second reason why we measure the genotoxicity of the fecal water by the COMET assay, which is the single cell gel electrophoresis assay. It's like this experiment is relatively, it's not too complex. And it is it's nicely to be develop a high throughput platform, and there are actually a high throughput platform available, and this platform can actually be used for like a large sample size measurement. Yeah, I think I answer your question.
Okay, thank you. So in the chapter 3. There's another questions. So from the findings of this study, which reviews the association between HR receptor activity and the intestinal inflammation. So as this receptor is also involved in the response to various environmental factors, such as exposure to heavy metal or pH, polycycloacrylamide hydrocarbons. So do you think that exposed to environmental chemicals play an important role in the occurrence and the development of IBD. So what's the contribution? And do you think these several factors like pH, AHR, and the dietary factors and the gut microbiota, what's the interaction between these factors?
Okay, so I think your first question is about like the environmental AHR agonists, such as the PAH, which is the polycyclic aromatic hydrocarbons. They are well-known AHR agonists, and they have a very strong AHR induction ability. But at the same time, and like previously, they were thought to be carcinogenic compounds, right? Yeah, I think I have discussed this this part of things in my review. So actually, like the environmental chemicals such as dioxin and dioxin-related compounds, and also like pH, they were reported to modulate the gut microbiome and also regulate the immune response. And even I think like one publications that mentioned that the oral exposure to the benzoapirin is a very well-known PAH and is very carcinogenic. And the oral exposure to the benzoapirin actually induce the gut microbiome dysbiosis. In addition, they induce like median intestinal inflammation. So, but I think I actually have already discussed like either high AHR activation or low AHR activation. Actually, both situation will induce the intestinal inflammation. So it is very challenging. So as I mentioned in the chapter 6, the page 181, and then I mentioned it's very challenging to determine the proper level of the AHR activation that required to achieve the protective and the therapeutic effects without side effects. Yeah. So I think like if we really want to apply this environmental AHR agonists as a potential, for example, therapeutic targets, the surveillance of intestinal AHR activity together with the intestinal inflammatory and carcinogenesis marker in population might be useful to improve the AHR targets treatments. Yeah.
Okay,
Okay.
thank you, Professor Wen
Thanks
Chen.
for your-
And we now turn to the next opponent, who is also the secretary of this degree committee. By the way, I want to apologize for the quality of the sound system, also for the audience, because sometimes it's very awkward actually, but we can't help that at this moment, of course. So the next question will be asked by Professor Daniel Castelli, and he's professor of gastroenterology in our university.
Thank you very much. Dear candidate, first of all, also congratulations from my part for this excellent thesis, and also congratulations to your supervisory team who have helped you in this work. I was particularly intrigued by the fact that you have looked at a clinically very relevant problem, diet and IBD. So that brings me to my very first question. If I were to see a patient this afternoon at the outpatient clinic, and if this patient asks me the question, what should I actually eat, doctor? What would then be the answer based on your findings and your expertise at this moment?
Highly esteem the opponent. Thanks for your kind words and your question. I think actually I answer this kind of, I give like a answer to this question based on the answer to the Dr.
Professor Black?
Yeah. So I think before you give any suggestion, you need to know like how is the gut health status of these patients, right? So whether there's like active inflammation present.
But for the interest of time, I'm gonna intervene here. So let's just assume that this is a stable IBD. There's no inflammation at all, whether it's Crohn's or colitis. So the patient is in endoscopic and biological remission. what would then be your advice? Because this is a question that we very often get from patients.
Yeah, for sure.
So it's a stable patient, no inflammation at this point.
Okay, but does this individual has any malnutrition?
No malnutrition, perfectly normal someone who is very, very health literate, so very invested in doing the right thing, but they want to do the right thing. So what would your advice be? Eat lentils all day or eat something different?
But I still want to know more details.
Okay, fair enough. So let's
because
let's
it's very
assume,
difficult, you know.
Yeah, fair enough. It is a hard question. That's why I'm asking you, because I cannot answer it. So the other thing that you mentioned with Professor Black is that this aspirin recommendation also says that during flares in particular, you should have a higher intake of protein than general. Is that correct?
I think based on the guidelines from the European colitis and
So the concern that I have, and in particular when it comes to tryptophan, you're suggesting that these tryptophan metabolites have beneficial effects. You say they exert protective and anti-inflammatory effects, but I also do have knowledge to the contrary. So for instance, indole acetic acid, but in particular, indole acrylic acid can actually also have detrimental effects because they have this highly reactive potential. That's why they're actually antioxidant. But at the same time, they can also react with just normal molecules like the epithelial cells. And there's just a paper that's now out. I think it was Nature Metabolism or something very fancy. Is that they showed that indole acetic acid can inhibit Paneth cell proliferation, so that's something that you would think is beneficial. So what's the right balance here with high proteins but not having any detrimental effects of proteolytic activity? What's the balance there? Short answer, please.
Sure. Okay. Yeah, I think this is actually the same question about like the AHR activation, because indole acrylic acid is the strongest. I mean, based on all the tryptophan metabolites, the indole acrylic acid is the strongest AHR agonist, actually. And it's the same as the environmental agonists, like a pH. They are like a very high AHR agonist, but at the same time, they could be detrimental to your health, right? Yeah. And we also need to mention that like the AHR-mediated effects on the gut health are very context and very context-dependent. So under the healthy status, actually, if you like highly induce your AHR activation, it will induce oxidative stress and also inflammation, as you mentioned. But under the inflammatory status, the activation of the AHR actually they will protect the intestinal inflammation. They will prevent and also show anti-inflammatory effects. So
Okay, so the short answer is we don't really know yet. Is that okay? Excellent. So I have one last very short question, which regards this perfect picture that you also showed in the introductory lecture, is on page 172 on the 3 different pathways of tryptophan metabolism: kynurenine, serotonin, and indole pathway. Now, the thing that concerns me if we are actually asking patients to eat more protein is that generally there's another enzyme that you didn't indicate in this figure, which is called the tryptophan deoxygenase, TDO, which is an enzyme that does not come into expression in the intestine. So your figure is absolutely perfect. No problems there. The only thing is that if you eat a lot of tryptophan, it will be absorbed by your intestines. It will go through the portal vein to the liver. And this TDO enzyme is something that is also induced by tryptophan intake, in addition to glucocorticoids, which is also an issue in IBD. So is that something that you would consider taking into account when we are actually advising patients to take a lot of tryptophan, either from lentils or whatever else?
Yeah. I think, first of all, the figures that you mentioned actually describes the tryptophan metabolism in the intestine. And the TDO is highly expressed in the liver. Yes. So that's why I didn't mention the TDO in my figure. That's one thing. And the other is, I have to say, to be honest, I didn't take into the liver metabolism of tryptophan into account into my study That's true. And I definitely agree with you. The metabolism of the tryptophan in liver must play a very important role. But I mean, based on my study, I didn't look into that.
Yes, thank you. I'm very, very satisfied with your answers, and I'll give the word back to the prorector.
There are always more questions, and that's the function of research too, huh? I now turn to Professor John Penders. He's a professor of medical microbiology at our university.
Thank you. Dear candidate, I would also, of course, like to congratulate you and extend my congratulations to your supervision team with this thesis, which I also read with great pleasure. But of course, I also would like to exchange thoughts with you. And maybe you or one of your paronyms can first read out proposition number 4.
The major AHR ligands in fecal water still needs to be identified.
Thank you. So I assume that this proposition is based on the results of chapter 4. Reading chapter 4, you first, and you also showed that in your presentation, showed that there is actually a positive correlation between tryptophan metabolites in fecal water and AHR stimulating activity, but also a negative correlation with fecal calprotectin. But when you then apply these individual metabolites, you cannot prove these findings. So apparently you trust one of your experiments more than the other. And I would like to know, why is that? So don't you find these associations to be true, or is there something else going on there?
Highly esteemed the opponent. Thank you for your kind words and your question. I think like these two results, they are not contradict to each other. Yeah, indeed, like the tryptophan metabolites are associated with the AHR activation of the fecal water, but it cannot explain like the AHR activation of the fecal water. So I assume like the production of the tryptophan metabolites, they are definitely related to the production of the major AHR compounds, like AHR modulators, right? And actually, we have already do some exploration to identify the major AHR agonisms in the fecal water. And our strategy is we just like run the liquid chromatography of the fecal water, and then we separate the eluent into, like, for example, 48 fractions, and then we use each fraction to treat the colonic epithelial cells, and then we measure the AHR activity, and then you can know like which fraction actually they induce the highest AHR activity, right? And at the same time, we also have the mass spectrum data of the LC measurement, right? And then you can always link back the fraction, like the cell experiments data, back to the mass spectrum data to identify like what is the potential group of compounds they are possible AHR agonists.
Okay, so that already answers one of my follow-up questions. So that's
okay.
That's great. But maybe go back to the set of indole metabolites that you use in this pool. You mix these 10 metabolites, right? And coming from the infant microbiome, I missed one of the metabolites, namely indole lactic acid. And I was wondering why you excluded that one, because it has been shown, at least for early life, that it's a very potent AHR activating ligand, but also has an impact on innate lymphoid cells, as mentioned previously, but even on adaptive immunity. So I was wondering why missing out on that one.
Yeah, I have to say, actually, I didn't measure all the tryptophan metabolites that could produced by the gut microbiome. But I mean, like previously, when I tried to select this compound, like the targeted compounds, I just look into like for example, like a Google pathways to try to identify like the key metabolites and the key pathways that involved in the tryptophan metabolism. And additionally, when we want to by these standards, not all of the standards were found. So I think that's the main point. Definitely, I agree with you. It's better to explore all of the tryptophan metabolites, right? Yeah.
And maybe, so you say you're now looking at different metabolites that might have a role in that, but what about the set of 10 that you tested now? Previously, you mentioned antagonistic and synergistic effects. Could it also be that just by pooling these, you average out the effects, and that there might be something going on that you now miss by creating this pool immediately of these 10 metabolites?
But I think like if you I think in the chapter 4 I think we treat the cells with the single compounds of the metabolites, but we also treat the cells with the mixture of tryptophan metabolites. So actually we explore like the single effects as well as their combined effects in the chronic epithelials.
That's a big leap, right? So it's from 1 to 10, but not like 2 at a time, and then making these matrices.
Yeah, that's true. But then I think I definitely mentioned like some publications. They have already like explored like the two like combination of two tryptophan metabolites on the chronic epithelial cells. But that's true. I didn't do that, and it would be interesting to do that in the future. Definitely.
Okay. I thank you for your answers, and I give the word back to the pro-rector.
Thank you, Professor Penders. Probably the last opponent is Dr. Misha Vrolijk, Assistant Professor in Food Toxicology at our university.
Thank you very much. Dear candidate, first of all, congratulations for this beautiful work. I think your thesis covers a very interesting field, linking diet to the pathology of IBD with the specific aim of providing more mechanistic insights. Being a toxicologist with an interest in food safety, that excites me. So it was really joyful and interesting to read your work. You did a lot of interesting studies, and as been mentioned before, you integrated a variety of methods in your thesis, and on top of that, most of your work is based on human data and human samples, and I think that adds a lot of relevance to your work. So my compliments for that, and with that I'd also like to address your supervision team. Of course, I'm here to ask some questions as well. When I received your thesis, I was immediately triggered by the cover of your thesis, where I see lots of vegetables, I see some fruits, and as you highlighted in your presentation, a healthy diet is associated with a decreased risk of developing IBD. Now, many of the intervention studies, dietary intervention studies, tend to focus on the macronutrients, so proteins, sugars, fats, and they tend to overlook the micronutrients. Well, we all know a healthy diet consists of fruits, vegetables, which are rich in micronutrients. So my question to you would be, what is now your view on the role of micronutrients in the development of IBD?
Like highly esteemed the opponent, thanks for your question. I would say, because you can see in the chapter 2, I actually look into the dietary factors into the several aspects. We include the food groups, and we also look into the nutrients, and we also look into a dietary pattern, right?
Yeah.
Yeah, and then I would say, in my opinion, I prefer to use a dietary pattern because it will give you the hints between the interaction between the different kind of foods, right? And this is, and also like this is like a common way that the human take. They will not specific like they will not only take into only take one specific nutrients or one specific food groups. Like normally they will take a pattern of diet.
Yeah, I agree with you. So you're talking about nutritional intake, which is on the one hand very important. On the other hand, you also have nutritional status. So we know that IBD patients have more difficulties with absorbing the micronutrients. So I want to use in my following question an example of vitamin B6. Vitamin B6 is an essential vitamin for many enzymes that are involved in the tryptophan metabolism. So that links very well to chapters 4 and 5, where you discuss a changed tryptophan metabolism. So if you have low levels of vitamin B6, your tryptophan metabolism will probably also affected by that. Could it be in your study population that differences in vitamin B6 levels explain the differences in the tryptophan metabolites detected? And would it be worth looking at the status of vitamin B6, for instance, in your study population?
Oh, okay. I think like so I think we have the nutrient intake data from the IBD patients and the healthy controls. And I think we also have the vitamin B6 in the data. Yeah, I think you can find the data in the chapter maybe chapter 4, I think. You can find the data in chapter 4. And I think there's no difference between the vitamin B6 levels among like different groups.
Yes, I saw the table, and I do agree with you. So there were very small differences between the intake of vitamin B6 between healthy controls and the IBD patients. It is shown in other studies that the status of vitamin B6 in IBD patients is lower than healthy controls. So despite the same intake of vitamin B6, you might have a lower absorption of vitamin B6 in IBD patients. It's also been shown that IBD patients in active disease state have even lower levels than in non-active state, so in stable state. So if we talk about, and you discuss a couple of times in your thesis, the potential therapeutic target of your tryptophan metabolism, do you think that vitamin B6 could play an important role there? So should we more focus on the micronutrients rather than maybe the macronutrients there?
I think I totally agree with you that vitamin B6 must be taken into account when we want to really study the tryptophan metabolism. That's a part that is missed in my thesis. But I think for the next step, if we want to do any like dietary intervention, like for example by applying the tryptophan metabolites or interacting with like the enzymes that are involved in the tryptophan metabolism, you need to take into the status of the vitamin B6 into account. Yeah.
I'm happy to hear that. Have you been able to check the vitamin B6 levels in the fecal water samples?
I think you can definitely do that, even though I didn't do that, but definitely you can do that.
Because I think that could already give a nice indication if there were differences.
Yeah, I totally agree, yeah.
Okay, thank you very much. Then, in chapter 4, you expose yourselves to fecal waters, and my previous opponent already started discussing a little bit about that. So your fecal water mixture is a complex mixture, so all kinds of chemicals are inside. But it's only looking at the water-soluble fraction, right? So is it fair to show the effects of fecal water samples on AHR receptor activation only by using a hydrophilic extract, or should you also look at a lipophilic extract?
I think there are two main reasons that I can answer this question. So for one thing, like all the compounds after the digestion from the gut, from the stomach till the colon, actually most of the compounds has been transformed and degraded to a more polar compounds.
But you can finish your answer. Shortly.
Yeah. So I would say like most of the compounds in the fecal water, they tend to be polar or medium polar compounds. That's one thing. And the other is we have to at the very beginning of my project, we isolate the lipophilic and also hydrolytic extractions of the fecal water, and we find the lipophilic extraction are very cytotoxic, and with non-cytotoxic concentrations, they induce very low AH activity. So that's why we stick to like a water-soluble extraction of the fecal water for most of my like all of my experiments.
Thank you very much.
Thank you. Shan Wang, the time appointed to the defense of your thesis has now passed, and this implies that the committee here will withdraw to discuss the quality of the defense and of your thesis. And I would ask you and your company to await the results of our deliberations here in this room, and thereby I adjourn.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Please be seated.
Sean Wang, the degree committee, here present, has assessed the quality of your thesis and of the defense that you just gave, and in view of its positive verdict and taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. Professor Van Schoten, your supervisor, is authorized to confer upon you this academic distinction in accordance with Dutch University custom. I invite you, supervisor, to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Sean Wang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. Congratulations, Dr. Wang. Dear Sean, I'm so proud today that you are here and you did so great. And I will give the floor to your supervisor, Rocher, for the laudatio.
Dear Sean, dear Dr. Wang, I'm happy that I am the next person that can congratulate you with your PhD. I would like to look at your PhD project as an exciting journey, with today as your destination. This journey started in China, in Longyan, where you were born, and from there you traveled to the University of Guangzhou City, which is the first part of the journey, which is 550 kilometers. In Guangzhou City, you met Professor Chen, and she was actually in touch with Frederick Jan. She asked Frederick Jan whether he would be willing to accept you as brilliant student, as PhD student in Maastricht. So we organized a Zoom session to meet each other. The connection was extremely bad, and you always had to ask, can you please repeat the question? This must have been very stressful for you, but also for us, because we said, should we really hire her? Right? She didn't understand the questions. But luckily, we did accept you, and you came to Maastricht in November 2018. That's a distance of 9,198 kilometers. So if we add that to the first trip, the 550, then we end up with a sum of 9,748. You started working in Maastricht at our department, but you didn't do this all alone. We had a very nice and fruitful collaboration with Daisy and her co-workers. I do not want to go into specifics because we discussed that enough today, but I do have to mention that we were always very impressed with meetings that we had with you. You knew what you wanted and you had a very strong own opinion. Actually, we just had to think along because you had everything already planned ahead. The quality of your work is also reflected in the fact that you won the poster prize at the annual meeting of the Dutch Society of Toxicology, the NVT, in Ede. Winning this poster prize also gave you the opportunity to present your data at an international conference, the EEMGS. Unfortunately, this meeting was cancelled due to COVID. But eventually, you could give your presentation at the next EEMGS, which was here in Maastricht. So for your PhD journey, I cannot add those kilometers, unfortunately. Actually, you visit the NVT, so the Dutch Society annual meeting, and the EEMGS twice. You also visited a conference in Edinburgh, and you plan to visit the SOT in the US. But also that one was cancelled because of visum problems. Yeah. So sorry, Sharon, also that trip I cannot add to your kilometers. So the total kilometers in your PhD journey so far is 9,748 plus 2,334 is 12,082 kilometers. Wow. You are also a real team player. In your project, you work together with Koen Venema in Venlo, and you worked with researchers in Amsterdam, Timo and Marja. You actually visited Amsterdam to do experiments there, and these experiments form the basis of the work that we are still doing here in Maastricht, and in which we try to find the actual AH receptor agonists. Traveling for these collaborations, so to Venlo, to pick up the samples for the TIM2 model, and to Amsterdam is a total of 604 kilometers, which brings your total now to 12,686. After your contract here in Maastricht, you went for a postdoc position in Baltimore in the United States. I think this will allow you to even further develop as a researcher, because you will not only be working on a completely new subject, but you will also experience differences in research culture. Now you have work in China, in the Netherlands, and the US. So I think by now you know the differences. Maastricht is the best, right?
Yes.
Okay.
Definitely.
So now you are in Baltimore, which is a distance of 6,564 kilometers. Initially, you wanted to do your thesis defense online, but I am happy that you decided to do your defense here on site so that we could meet face to face again. As a result, I have to add those kilometers from Baltimore back to Maastricht to your total. Oh, I forgot some kilometers. Here in Maastricht, you lived in a nice apartment close to the railway station. This was your base from which you traveled to work and back. But you also visited a lot of places here in Europe. In a journey, it is not the travel itself, it's also not the destination, but what's most important is the people you travel with. You traveled a lot with your friends, colleagues, and family. And some of them are actually here in the audience. I think you enjoyed your stay here in Maastricht, but you certainly enjoyed your travels, because I saw some wonderful pictures that you took from all the cities that you visited. However, all those fun trips, I cannot add that to your kilometers, right? The only I can count is that you went from your apartment to the university and back for approximately 5 years, every week, every working day, and sometimes in the weekends. So if I add up all those kilometers, I come to a total of 29,124. This is a very special number, 29,124. That is today.
Wow!
The 29th of January, 2024. Yeah. So this is a very special day, and this is your special day. I wish you good luck in the future, but first, enjoy today. Congratulations.
Thank you. Thank you so much.
There was a picture?
Yes, the picture.
One more.
One more. Okay. Yeah, thanks.
It'll travel around the world, this picture, it will. Dear Dr. Wang, on behalf of the academic community here in Maastricht, I would like to congratulate you also with the degree you just acquired. And of course, I extend congratulations to the supervisory team, to the Department of Toxicology, to the School of Nutri, to all the friends, family, those online. I'm not a specialist in this kind of thing, but I did read the summary and, well, you know, sort of leafed through it. And of course, I listened carefully to all the opponents today and what they had to say about the work that you did. And I think everybody was impressed with the quality of the work. It has been a productive project, I think. And since we had this terrible COVID epidemic during the period that you were here in Maastricht, I think it is also an accomplishment that you did this within this period, within 5 years at least. So there was some speed as well. It's quality, but it's also speed and quantity. And the speed was visible today as well in word production. You told me you were a bit nervous, but it didn't show too much, I think. So congratulations with that too. And I think, you know, having a postdoc position in Baltimore is also a recognition, I think, of the work that you did here and the quality of that. Because not everybody is invited or selected to take up a position over there. I wish you all the success in the world over there. Having said all this, I want to thank the opponents for their contributions, and also, of course, especially those who were here online, guests, and guests always indeed are also a testimony for the quality of work, because they were interested enough to take the time to contribute to this session. And having said all that, I come to some of the household announcements. We will take a picture here with the opponents from elsewhere as well on the screen. Before we do that, and to make it a bit of a quiet surroundings, I would ask you to already leave and maybe go to the Refter, to the reception area, where you can already take a drink. And then after a while, we will join you and you will be allowed to congratulate the new Dr. Wang. And thereby, I close this session.
Thank you. I think so. Did you have a good idea?
Recording stopped.